Welcome to today's lecture. Today's topic is fermented milk products. So in this topic we will discuss about what is fermentation, the process of fermentation, little bit about cultures, that is started culture, and also different kind of fermented products starting from dahi, yogurt, lassi, then lactobacillus milk, probiotic fermented milk, then about Srikhand, Kefir, Kumis and cheese. These are the different kind of fermented milk products, of which cheese is very famous in the western countries and the dahi is very common in our Indian homes, but the better version of it is yogurt. So all these things we are going to discuss briefly in this lecture today. So first let us understand what is fermentation. Fermentation is a metabolic process in which an organism converts a carbohydrate such as starch and sugar into an alcohol or an acid. So actually it is a process for the multiplication of the organism. It needs energy and that energy is produced in this process of metabolism and it produces either alcohol or acid. Commonly yeast plays an important role in fermentation of sugar into alcohol to obtain its energy, and that is the common process for making alcohol in breweries. Similarly, many bacteria can ferment carbohydrates and convert it into lactic acid, and in that process it gets the energy. In case of milk, lactose is the main carbohydrate and which is fermented by some group of organisms and they produce lactic acid in the process. So such organisms are called as lactic acid bacteria, and this kind of fermentation is called lactic acid fermentation or lactic fermentation. So we will see more details about it. Here let us understand about the importance of fermentation. It is well known that fermented foods are good for health. How it is good? Like it can preserve the food due to the formation of acid. In generally in case of food, fermentation produces acid. Then it enriches the palate of flavor. Due to this fermentation, it produces some flavorful components, allows a better assimilation and digestion. Because of the metabolic activity of the organism, so many complex compounds are broken, mainly starch or other things, and that becomes easy for digestion or assimilation. And also many of these fermenting organisms, especially lactic acid bacteria, are many times good for our health, particularly in our intestine. So that is how fermented food, any fermented food or milk products are good for health, and it is always preferred by us. Here is about the fermented milk products. As I mentioned at the beginning, in Indian condition or in Indian subcontinent, dahi is a very common fermented milk product. It is mostly done in home, but nowadays it is also available commercially. And in Western countries, cheese is the most popular fermented product. Then other important fermented products are yogurt or Srikhand is getting very important in Indian market. Then cultured buttermilk, Acidophilus milk, Kefir and Kumis. These are also important fermented milk products which I am going to discuss today. Now let us understand about fermented milks. A fermented milk is prepared by heat treating the milk to kill undesirable microbes and inoculating with a little bit of sour milk from the previous batch, which acts as a starter for the fermentation. So this is the normal process followed in home. First is the milk is heated, which kills the pathogen and other organism mostly, and then the old fermented milk is used as a culture for inoculation and which gives the fermentation for the new product. The fermented common milk products, they will have longer keeping quality due to the acidity. It will have a pleasant flavor and will have much safer to the consumer, because pathogenic bacteria have been killed and contamination with pathogens afterwards cannot lead to growth of these organisms. So firstly pathogens are killed. Secondly, once the fermentation started, if at all any contamination happen with pathogen, that cannot grow. Now let us see about the fermentation process. The lactic acid bacteria alter the conditions of milk in such a way that most undesirable organisms cannot grow or will even die. These conditions include a low pH, that is 4 to 4.6, a low redox potential, and growth inhibition by undissociated acids, especially lactic acid, and other metabolites such as hydrogen peroxide and compounds with an antibiotic activity. So due to the process of fermentation, the pH of milk goes down, and a kind of anaerobic condition, that is the negative redox potential, comes, and some inhibition of growth by lactic acid and other compounds like hydrogen peroxide or antibiotic properties prevents the growth of all other organisms except the starter culture bacteria. Pasteurized and homogenized milk is inoculated with an aromatic starter culture and incubated at around 20 degrees Celsius for 17 to 24 hours. So these are the common process of fermentation. So here briefly we will see about the starter culture, the culture which is used as a mother for the fermentation. Nowadays these are commercially available, as you can see in the photograph. Different commercial company, large scale, they are producing and supplying ready use, especially freeze dried powders for direct use. Generally the mother culture can be used for propagation by transferring a small amount of it in the growth vial or media or other containers, and it can be increased in large scale, and that can be used for actual application. So nowadays they are readily available in small size pouches or sachet for the commercial use for production of different kind of fermented milk products. Here is just a quick view about some of the common appliances which is useful to see for the undergraduate students. You can see the beaker where it is a conical flask where the milk is inoculated and plugged with cotton for propagation. We can see the autoclave, which is very important for sterilization of glasswares especially. And the last right side we can see the incubator, which is used for incubation of any bacteriological purpose or for incubation of fermented milk products, especially for yogurt. We can fix a suitable temperature around 41-42 degrees Celsius for perfect fermentation. Here we will see the most common process of making dahi, which is done in every home in India. So the brief procedure is, first milk is to be boiled, then it should be cooled down to around 40 to 45 degrees Celsius. After that, 1 spoon of old dahi from previous day or from other source can be used and mixed thoroughly, and then we can close it lightly and leave it for fermentation. So this part for leaving fermentation, that is very important. It should not be disturbed, and the temperature is very important. So in the normal, most of the part in India, the temperature is above 20, so 20 to 35 degrees, okay. Only in the winter, some part of India, in the northern India, the temperature could be 10 degree or below, so that time it is difficult. Otherwise the curd can be made anytime easily in home. Here we will discuss some of the common problems faced in making curd in home, like sometime it can be not set properly, or it may have loose body, it may be extremely sour, or sometimes slightly bitter, or sometimes there will be watery top surface. So the reasons are, like if it is not setting properly, maybe the culture is not proper, or maybe it was added when the milk was too hot, though some of the organisms died. If there is loose body, again either due to the culture not working, or maybe too much of water present in the milk. Sometime if it is extremely sour, maybe the culture is more and it has got over fermentation, or the time given was more. Sometime not set properly, it could be due to improper temperature or the too cold climate. Sometime we can get the surface water too much, that could be due to the milk which is having extra added water. So these are some of the common issues happens in case of curd in home. Now we will discuss about preparation of curd in the dairy, that is in the dairy plant, where it is done more scientifically. So first milk is taken, then it will undergo filtration, then there is a preliminary heating up to 65 degrees Celsius, then homogenization. So many times it is important to mix everything thoroughly, especially in the dairy plant they will add extra milk powder to bring better thickness or texture in the curd. After homogenization, there is again further heating or pasteurization around 90 degrees Celsius for 5 minutes. Then it will be cooling to 42 degrees Celsius. Then inoculation with starter culture about 1 to 2% at 42 degrees Celsius. Then incubation around 40-42 degrees Celsius till the acidity reaches 0.95 to 1%. So this acidity can be checked after 4 hours, and by this time it will be setting. There will be thickening and coagulation. So it becomes a set dahi or curd. After that we can stop the further fermentation by removing it from the incubator and keep it in the refrigerator. So when we keep it in the refrigerator, temperature is around 5 degrees Celsius. So the microorganism, the starter, cannot grow anymore. So acidity will not increase and it will have a better flavor and acceptability. Here few more things we will discuss about the milk for making curd. Firstly, it should be produced hygienically, then only we can get good quality and safe products, and it should have good quality raw material to get the better flavor. Fat percentage approximately 4 to 5% in the milk is preferable. Then higher the total fat or total solid, higher the firmness or lower syneresis. I have mentioned earlier, sometime a 1 or 2% up to 3% milk powder or skim milk powder is added for better thickening and texture or firmness. So total solid minimum it should be 12%, and sometime if we add milk powder it will be still better. Now this is briefly about heating of milk to 90 degrees Celsius for 5 minutes. This is for pasteurization and making the milk safe and perfect for fermentation to increase the recovery of whey protein in the product and increase the yield. That temperature is very good. And the cooling of heated milk to 42 degrees Celsius, because at this temperature the starter culture remain active and it can easily convert lactose to lactic acid and perfect setting of dahi. Here we can see commercially available dahi with different brand and different packaging, different form. It is a old practice of making dahi in earthen pot. Nowadays we can see the polystyrene cup or sometime LDP pouch by different company. It is commonly available for marketing of dahi in Indian market. Here we will see the preparation of lassi. So lassi is prepared from dahi. It is a very common drink in northern part of India. So it is processed very easily in home and also in many small restaurants. So firstly the dahi is taken, then they will break the set curd and it is mixed with water or whey, and the sugar or any other sweeteners are added. Then some flavor is added and then it is mixed thoroughly with some mixie or some homogenizing devices, and then it is filled in some cup or bottle and kept in refrigerator and it is served chilled. So this is a common preparation from dahi, that is called lassi. Now we will discuss about yogurt. Yogurt is also a kind of curd, but it is a special kind of curd with very specific culture. So yogurt is produced by the controlled fermentation of milk by two species of bacteria, that is Lactobacillus bulgaricus and Streptococcus thermophilus. The sugar in milk is called lactose, which is fermented to lactic acid by Lactobacillus bulgaricus, and it is the one which caused the characteristic curd to form. So yogurt is a special kind of curd where specific cultures are used and they need a very specific temperature, and it gives a typical flavor and other advantages. Here in yogurt we will understand how these two specific organisms grow together in a symbiotic manner. Firstly, Streptococcus thermophilus with their growth bring down the pH of milk to 5.5, and at this pH it is suitable for the growth of Lactobacillus bulgaricus, and which produces proteolytic enzymes. These proteolytic enzymes causes proteolysis of milk protein and peptides are formed. Now these peptides stimulates the growth of again Streptococcus thermophilus, which in turn produces other acid like formic acid and carbon dioxide. Now these two components further help in the growth and stimulate the growth of Lactobacillus bulgaricus. That is how these two organisms alternately help each other and continue their growth and fermentation proceeds. Here we will see the different types of yogurt like steered yogurt. Here after fermentation, though there is a setting, but it is broken and by stirring and mixing. Then drinking yogurt, here the solid content at the beginning itself very less and it will be liquid even after fermentation. So that is taken as direct drinking yogurt. Flavored yogurt where special flavor is added. Concentrated yogurt where the extra milk powder is added, so it will be very thick and solid. Then frozen yogurt in freezer after the fermentation, and then set yogurt. Here after fermentation it will be undisturbed and it will remain semi-solid and set. So these are sometime nowadays they are making fruit yogurt. In the milk itself different kind of fruits are added. So these are different kind of yogurt. Here is the process for making yogurt. The process is almost similar what we have learned for the preparation of curd or dahi. Only thing is here the culture is very specific and here the some fruits or sugar or specific starter culture is used. So first the raw milk, it is heated and pasteurized. Then pasteurized milk, sometime we can add some milk powder, so there will be homogenization and cooling, and then the starter culture is added at a specific ratio. Then it is kept for incubation. The temperature of incubation is very specific here, that is 41 to 42 degrees Celsius, because these two organisms which I have mentioned, they are partially thermophilic, so the temperature of 42-43 is ideal. After this incubation, within 4 to 6 hours, the yogurt will be ready. So that is raw yogurt. Nowadays, over that, sometime fruits or flavor can be added, or sometime the flavor is added at the beginning itself, and sometime it is further heat treated to make processed yogurt. Now we will talk about natural buttermilk. So this is a byproduct of churning cream for butter making. Ripened cream which has undergone a clean lactic fermentation is usually preferred. So earlier we have discussed about the preparation of butter, which is normally prepared from cream. Now this cream, if we allow it to ferment and keep a separate period of ripening, then it is very good for making better quality of buttermilk. So this is coming from normal preparation of butter. But in the home level, there is a natural buttermilk prepared at country level, that is called country buttermilk, which is also obtained as a byproduct of churning whole milk or curd for production of country butter. So in home level, many times they will make butter from the fermented milk or curd. At that time we will get a buttermilk, that is also a byproduct from butter at the home level. So these are the natural buttermilk. Now we will discuss about cultured buttermilk. Cultured buttermilk means when the cream is specifically fermented with some starter culture, and from that when we make butter we will get the buttermilk, that is cultured buttermilk. Cultured buttermilk is the fluid remaining after ripened or sour cream is churned into butter using mesophilic starters. Different products are produced by using different strains of lactic acid bacteria as starter. Butter is normally made by churning cream that has been soured by lactic acid bacteria. So sour cream uses different kind of cultures like Streptococcus cremoris or Streptococcus lactis for producing lactic acid, and Leuconostomus cremoris for characteristic flavor. So these are some of the culture which is used for fermenting the cream before churning. So cultured buttermilk means when the starter culture is specifically added on the cream before making the butter by churning, and the byproduct liquid we get is the cultured buttermilk. So here is a composition of buttermilk gram per 100 gram. So most of the total solids 9.5 to 10, and there is a comparison between conventional buttermilk and cultured buttermilk. There is almost similar. In case of conventional, the fat content is more, that is up to 3%, whereas in cultured buttermilk it will be very less. Then protein is around 3 to 3.9 in both the case. In lactose, almost all the lactose will be in the buttermilk, because lactose normally remain in solution form in the milk, so that fully goes with the buttermilk. And lactic acid around 0.5% in both the case, but the concentration can be lower due to addition of water. So how much water add, that decides the concentration or solids. Similarly, concentration can be changed by or increased by addition of skim milk powder. Here is another type of buttermilk, that is Bulgarian buttermilk. Bulgarian buttermilk is a high acid fermented milk made from pasteurized whole milk inoculated with Lactobacillus bulgaricus alone at a level of 2% inoculum. So earlier we have seen Lactobacillus bulgaricus and Streptococcus thermophilus together used for yogurt. Here only Lactobacillus bulgaricus is used, and the fermentation is done at a higher level. So here, after putting the culture, incubation is done at 38 to 42 degrees Celsius for 10 to 12 hours until a curd forms with about 1 to 2% acidity. The product has a sour flavor and is popular only in Bulgaria. So here fermentation is done only one culture, and it is allowed to grow for longer period. Naturally acidity will be high. Now from this, when we make the buttermilk, that is called Bulgarian buttermilk. Now we will understand about Acidophilus milk. So Acidophilus milk is cultured with Lactobacillus acidophilus, whose primary function is to produce lactic acid. Here the sterilized milk is inoculated with a large amount of starter, that is 2 to 5%, and incubated at 38 degrees Celsius for a long time, that is 18 to 24 hours. So naturally the starter culture is high level and time given is high, so the acidity will be very high, so it will be very sour. So because Lactobacillus acidophilus is fairly acid tolerant, the lactic acid content of milk can become high, that is up to 1 to 2%, because this culture can grow even when the acidity is high. So that's why the final acidity can be up to 2% and it can be very sour. This acidophilus milk has two important issues. One is because of the high fermentation and acidity, the flavor of the sour milk becomes too sharp, and due to this high acidity, slowly the bacterial cells will be reduced. That's an important problem with the acidophilus milk. Now this problem can be overcome by blending plain milk with a deep frozen concentrated culture of Lactobacillus acidophilus, and by keeping the mixture at low temperature, say around 4 degrees Celsius, which prevents the milk from souring. So this is one of the different aspect of the acidophilus milk. So there is another new variety, probiotic fermented milk. So here the milk is fermented, plus it is fortified with extra probiotic, which is having excellent health benefits. But probiotic means it has to survive in the stomach acidity, then only it can give the benefit in our intestine. So there are specific culture, organism which has got probiotic role, but along with this we have to use the starter culture. So in this case, Lactobacillus acidophilus will be there as a fermentation, and additional probiotics will be added from the species of Bifidobacterium or Lactobacillus, some specific strains like Lactobacillus casei and Bifidobacterium bifidum. These are the most commonly used probiotic bacteria in the manufacture of fermented milk. So here normal starter culture will be there, in addition probiotic will be there. So then it will give a special health benefit. So it becomes a functional dairy product, that is probiotic fermented milk. Now here is another important Indian sweet dish from the fermented milk, that is called Srikhand. Srikhand is an Indian sweet dish made from strained yogurt. So yogurt is strained and liquid is allowed to go. After that sweet is added, even the fruits are added, and then it is kept in chill for serving. So that's the Srikhand. To prepare Srikhand, yogurt is tied in a cotton cloth and left under pressure to drain. The strained yogurt, referred to as chakka, and sugar are mixed thoroughly in a deep bowl. Sometimes we can add dry fruits like cardamom, saffron and any other flavors, and then added and mixed. It is then left in the refrigerator for the sugar to dissolve, and the dish is served chilled. So this is the specific sweet dish prepared from yogurt or curd after filtering and removing the liquid part, and special flavor or dry fruits and sugar is added and mixed and served chilled. Now we will learn about kefir. This is a very popular product in West Asian countries. Here during the fermentation, lactic acid and alcohol are produced. So normally we have seen for all fermentation in dairy it is the lactic acid, but here it is producing the lactic acid and also alcohol. So it is mostly fermented by yeast and also lactic acid bacteria. The microflora of kefir is variable. We will have lactococci, that is lactococci lactis or cremoris or sometimes diacetylactis. Then leuconostosis, that is leuconostosis lactis or cremoris. And then lactobacilli also can be there, like lactobacillus brevis or lactobacillus kefir. Also sometimes lactobacillus bulgaricus and lactobacillus acidophilus. So they all form the lactic acid bacteria, but along with that there will be the yeast which produces the alcohol, which we will see the next slide. So in this kefir production, I said there are some yeast culture. Those are like Candida, Pluiveromyces and Saccharomyces. These are the common yeast which are used in the kefir, and they produce the alcohol, whereas lactobacillus and lactococci species, they produce the lactic acid. Here the lactic acid content can be up to 0.7 to 1%, and the alcohol content can be 0.05 to 1%. So this kefir is having both lactic acid and alcohol, a special kind of fermented dairy product popular in West Asian countries. Now we will see one more fermented popular dairy product from Russia, that is called Kumis. Here also it will have both lactic acid and alcohol fermentation but it is done in two phases. In the first phase, lactic acid production by first phase of incubation by lactic acid bacteria, and in the second phase yeast is added and again incubation is done, and it will have a very high level of alcohol. So this is a lactic acid alcohol fermented milk. Earlier days it used to be made from mare milk, now it is from cow milk. It is originated from Russia, and the first phase the culture will have Lactobacillus acidophilus or bulgaricus. Then the incubation will be done, and in the second phase yeast will be added and again incubated. The finished product may have up to 2.5% alcohol. Now we will talk about the last one of fermented dairy products, that is cheese, which is very popular in Western countries, and cheese itself is a very long process to prepare. So we are going to have a separate details lecture on this. Today I will talk very briefly about the cheese. So there are different kind of cheese. I will just tell the kind of cheese and the example, like soft cheese, camembert and feta, semi-soft cheese, gouda and port soft, hard cheese like mimolette or pelorina, semi-hard cheese that is cheddar or cantal, and then fresh cheese like cottage cheese or mozzarella cheese, then blue cheese that is the mold ripened cheese that is Roquefort or Stilton, and there will be processed cheese. So these are different variety of cheese. Here we will see the different kind of cheese and the corresponding starter culture used. In case of soft cheese, Streptococcus cremoris and Penicillium camemberti. In case of semi-soft cheese, Lactococcus lactis or Brevibacterium linens. In hard cheese, Lactobacillus casei or Lactobacillus plantarum. In semi-hard cheese, Lactobacillus casei, Streptococcus cremoris. In fresh cheese, Streptococcus species. In blue cheese, Penicillium roqueforti or Lactococcus lactis. In processed cheese, fungi or fungal spores are used during ripening. So these are the different kind of cultures used for cheese making. So here I will briefly explain about the process of making cheese. So as usual, first milk should be taken, it has to undergo the pasteurization, then it has to be cooled, and then starter culture is added. I have told all the names earlier. Then some addition of coagulant like rennet. You have heard rennet is used, then that cause coagulation. After that there will be cutting of the coagulum, and then there will be draining of the whey and cooking of the coagulum. After that there will be kneading of the curds, then there will be salting of the curds, and there will be pressing of curd, and then finally ripening of cheese. So that is the very brief process for making cheese, but actually every step takes a lot of time. So preparation of cheese is a very lengthy process. I am going to explain it in full details in a exclusive separate lecture on this preparation of cheese. Now we are at the end of today's lecture. So today our topic was fermented milk products. So we have discussed first about fermentation and process of fermentation, starter culture briefly, and then we have discussed different kind of products like normal curd at home, and then dairy plant, how the curd is made, then about yogurt, then fermented milk with lactobacillus, and then we have seen other kind of fermented milk product like Kefir, Kumis, even Srikhand, the Indian sweets, and also we have learned briefly about the preparation of cheese. So altogether this fermented dairy products is very healthy. I have mentioned in some points, because of the effect of different starter culture and their enzymes, the proteins are broken, so we get small peptides which are good for health, and also it is easy to digest or assimilate. So fermented dairy products are very popular, and some of them are very old practice in different parts of the world. Thank you for listening.